start the class with something new, and then we will take it up in the Friday class. Okay, let me share my screen. What happened? Many people have not joined. Slowly, people are joining. Okay. Devanshi, I don't see her.
So she's probably just joining. She's actually been having network issues the entire day. So
Okay, okay. Because the earlier it was not like that, but nowadays MS Teams also gives you the attendance, and so I can't be marking somebody who is not there in the Teams as present. That's my problem.
so let me message her.
Uh,
I
no,
she
let it. Is there a group, common group that we can message or something? I
yes
know
sir, we
that
have a comment.
you have a common group. Okay, okay. Just alert everybody to join. Okay. All right. Let me share my screen, and then we will go. All right. Is my screen visible?
Yes sir.
Okay. So one thing that I inadvertently missed out last time was that when we do this, alpha complementation, or which is also called as blue-white selection, you generally see the entire purpose is we need to drive the beta-galactosidase gene, right? And what is the substrate for beta-galactosidase? It is lactose, right? We don't generally use lactose in the medium, but instead we generally use LB broth, right? LB broth is a rich broth. It has all kinds of nutrients in it. Okay, but somehow we need to induce the LAGZ gene, and so people typically use a chemical called IPTG, or which is isopropyl beta D thiogalactopyranoside. This is a non-hydrolyzable analog of allolactose. Okay, lactose gets metabolized into allolactose, and allolactose is the one that is actually the inducer for lacoperon. Okay, and so this is an analog of allolactose. So this acts as an inducer for the LAGZ gene, which we typically use it in the cloning vectors. Okay, so typically in when we do this blue-white selection, you know you would have both xgal and IPTG in it. Okay, IPTG is for inducing the LAGZ gene expression so that the beta galactosidase gets expressed, and this beta galactosidase is the one that is going to metabolize xgal or break down this xgal into this bright blue compound, by which we are able to know whether the your LAGZ gene has it been disrupted, has the reading frame been disrupted, so that if it is not disrupted, it is going to give you a blue colony, and if it is disrupted, then it is going to give you a white colony. Okay, so what happens if you don't add IPTG? What will happen if suppose you plate in an LB plate with just xgal, but you forgot to add IPTG? Any idea what will happen? Am I audible?
Yes, sir.
Okay, so what will happen if you suppose a researcher forgot to add IPTG, but instead added only xgal? What will happen? So in the absence of IPTG, your beta galactosidase will not be produced, right? Because that is the inducer. Okay. So in the absence of IPTG, the beta galactosidase will not be produced. So even though you have xgal in the medium, all the colonies will still be white in color. Okay. So it will look as though every colony is a recombinant one. Okay. On the other hand, suppose you added IPTG, but you forgot to add xgal, the result will again be the same, right? The result will again be the same. that you did not add xgal, and instead, everything, you have IPTG induction is going on. Even there, typically what will happen, because this enzyme is present, but the substrate is not there, right? Substrate is not there. You will still see only white colonies. Did I make it clear? So irrespective whether you forget to add IPTG or XKL, right? Both are needed. Both are needed. If you forget to add even any one of them, your plate will be full of white colonies. But you're going to get into these things. You may not know which is the actual blue colony and which is the white colony. Okay. Okay. So typically this plate will have both XGAL and IPTG. Okay, only when you have both XGAL and IPTG, this blue-white selection works. All right. Did I make it clear?
Yes, sir.
Okay. Very quickly, we will look at, in today's class, what shuttle vectors are and what expression. We have been focusing on cloning vectors, right? So far, we have talked about, cloning vectors. The most typical, the most common, widely used cloning vector is PUC. UC stands for University of California, and P, small p stands for plasmid. Okay, this is the most commonly used cloning vector. Okay, it has been replaced by a few other vectors, but if you look at it, most of them were pretty much the same way. Okay, so PUC 18, 19, they're still the most commonly used cloning vectors. Okay, so today we'll look at what shuttle vectors are and what expression vectors are. Okay, so if you see this picture, this should be familiar to you ideally. Okay, but I'm not sure whether you will remember it or not. But if you look at this is your typical cloning vector. Okay, and this cloning vector carries three components, which is your LAGZ gene. Okay, with multiple cloning site, which is also called as polylinker sites, where you will be actually be cloning your gene of interest. Your foreign gene will be cloned somewhere in this multiple cloning site. Okay, it has origin of replication. Okay, many people write it as ORI, but some people, when you go into the actual literature, they will say as REP. REP stands for replication protein. Okay, this is derived from again from a plasmid called PMB1. Okay, so this REP gene is from this plasmid. And then it has a beta lactamase gene which codes for ampicillin resistance. So this is typical of a cloning vector. Okay, and PUC18 is a very, very common cloning vector for cloning genes in E.coli. Okay, which is the most widely used organism in the academic laboratories. Okay, some people used to use Bacillus. Okay, but somehow Bacillus is gram positive, E. coli is gram negative. Working with gram positive things is a real challenge, and people developed a lot of things for Bacillus, but at some point E. coli just simply became, one of the most favored organism in all the academic laboratories in the world. In not only academic laboratories, it is a common tool even in commercial companies. Okay, so that's the structure of a cloning vector. Okay. In contrast, these two vectors I had showed you, these as expression vectors. So these two are called as shuttle vectors. Okay. And shuttle vectors can be very broadly classified as shuttle cloning vectors or shuttle expression vectors. So these two are shuttle expression vectors. I'm just showing you an example. Okay. Now, what is the principal characteristic of a shuttle vector? Okay. Anybody who can say this very quickly? I mean, looking at the picture, you will be able to see things. Any idea what are the two very in outstanding characteristics of a shuttle vector? Both PS2 and PIRES1 is a shuttle vector, and PUC18 is a cloning vector. Okay. Any idea what is the biggest this thing between this shuttle vector and the cloning vector?
Sir, antibiotic resistance selection.
No, you also see antibiotic resistance selection here, na? Cloning vector में भी है ना? You're almost there. You're almost there.
2 NCS.
Not really, because you are having only one MCS here, you know. This is also a shuttle vector. All of you are like, you know, all I can say is it is pretty much there in the picture. And they're all lying next to each other. So you will know, if you concentrate on one and compare it to the PUC 18, you'll be able to get it. Anybody else? It is so simple, you know, and then you will see, oh my God, okay, you know. Okay, nobody wants to guess? Yes. There are no, you know, marks or anything for guessing. You can guess. You people are right there, you know, antibiotic selection markers. You have to say it little more, you know.
It's the presence of poly.
Presence of poly A, no, that is only for mammalian. No? You don't see that here. Okay, let me show. Yeah, tell me.
F1 oris.
F1 ori. You are again, you know, you have pointed out something that is the, you know, few more things are needed. You are right there, you know.
The presence of two
Very good. Who is this?
Satanvi.
Tanvi, very good, beautiful. Okay, so what you see in shuttle vectors is the presence of two selection markers. One is ampicillin, one is URA3. This is for yeast. And one is neomycin resistance, one is ampicillin for in the mammalian vector. So in mammalian systems, you know, when you have to do this, you will be selecting for neomycin resistance. But in E. coli, when you do this in E. coli, you will be selecting for ampicillin resistance. Similarly, if you look at ORI, you know, you people are very close, you know, you will see multiple origins. Coli1 origin is there, SV40 origin is there, okay. Similarly, here you can actually see F1 origin is there and 2 micron origin is there, PUC origin is there. So both this origin and this origin, F1 origin and PUC origin, will work in E. coli, right? Similarly, here you have coli1 origin and F1 origin. These will work in E. coli, and SV40 origin will work in mammalian cells. In this case, 2 micron origin will work in yeast cells. So what you have is multiple ORI, or, you know, origin of replication, and you have multiple selection markers. This is the typical characteristic of a shuttle vector. Okay, did I make it clear? So you people are very close, you know. All you needed to do was to concentrate and see this. Okay, all right. So a shuttle vector is generally a plasmid which is constructed so that it can propagate in two different host species, right? For example, the PS2 shuttles between E. coli and yeast, okay, and the PIRES plasmid shuttles between E. coli and mammalian cells, okay. So therefore, any DNA that is inserted into this shuttle vector can be tested or manipulated in two different cell types. You know, you could clone it and then you could do the expression studies in the mammalian system. It's easier to clone things in E. coli and grow them up in high, you know, large quantities rather than doing it in mammalian cells. It's a difficult proposition to grow mammalian cells the way we grow E. coli. Okay? So that's why a shuttle vector is used. Okay, the main advantage of these vectors is that they can be manipulated in E. coli and they can be used in a system which is little more difficult than E. coli. It could be yeast, it could be, you know, mammalian cell, it could be insect cells, it could be any other cell type. Okay, so shuttle vectors are plasmids that can propagate across species. You know, some of these shuttle vectors can propagate between eukaryotes and prokaryotes, some between two different prokaryotes. For example, you know, there are certain plasmids which are useful in this species of bacteria called Rhodococcus erythropolis. Okay, And this is a different bacteria. So, you may need to do certain modifications in E. coli, and then you can go back and do all the manipulations in Rhodococcus. See, Rhodococcus is a very difficult to grow bacteria, unlike E. coli. Okay. So you can't be doing all the cloning and manipulation, but instead you can do almost all the steps in E. coli, and then you can transform Rhodococcus and see the gene function. Okay, so that is why this is called as a shuttle vector, which shuttles between E. coli, two different bacteria. Okay. There are also adenovirus shuttle vectors which can propagate in both E. coli and mammals. Typically, the plasmids that are used for deriving adenovirus vectors for your coronaviruses, right? Your this thing, they are actually the initial clones are made in E. coli, and then the cells get infected, mammalian cells get infected, and they then start putting out this recombinant adenovirus with a spike protein on their surface. Okay. Okay. So why are shuttle vectors? What is the advantage? Shuttle vectors are frequently used to quickly make multiple copies of a gene in E. coli, so that you can have amplification. You can make many more copies of it. Okay. And then you can use it for in vitro experiments and modification. For example, when you want to know, for example, if you see a protein with 1,000 amino acids, not all 1,000 amino acids are necessary for protein function. You could actually, we will see in applications of molecular biology how you can do protein engineering with the genes that have been cloned. Okay, so you can basically start removing certain residues and find out whether those are responsible for the function or not. Or you could do simply mutagenesis, right? You want to know whether a particular residue is there in the active site or not, you can go ahead, mutagenize, and do your studies. So that is the reason why people use shuttle vector. And as we saw in the previous slide, shuttle vectors generally have at least, okay, two origins of replication. One which is responsible for replication in E. coli, and the other one, it could be yeast, mammalian cell, or any other host. Okay, depends on the host that you're going to use. And shuttle vectors generally have two selectable markers, but in some cases, one may suffice. It depends on the host. Between, if it is not E. coli, if it is some other host, then you could probably get away with one selectable marker. Okay, so it depends. Okay. So did it make it clear, shuttle vector?
Yes, sir.
Okay. So once you have cloned a gene, right? Once you have cloned a gene and you say you now have a novel gene, right? You want to do expression of that gene, right? So the question is, where do you want to express your recombinant gene? Okay. Many different systems are available. You could clone and express it in E. coli. You can clone and express it in yeast. You can clone and express it in insect cells. You can clone and express it in mammalian cells, right? You have a variety of expression systems. Unlike cloning vectors, expression vectors, as we saw, are little different, right? In cloning vector, you're only able to say whether the gene got cloned or not. They don't express. They express very, very teeny, tiny amounts, okay? And which is not feasible commercially, okay? So these expression vectors are specially designed, okay? And the question is, so where do you want to, express your foreign protein, okay? So the most common thing, the first people attempt is to clone it in E. coli and express it in E. coli. So the question is, why do you want to express it in E. coli, right? Just imagine the case of Taq polymerase, okay? Any idea what organism this Taq polymerase comes from?
Thermus aquaticus.
Very good. Thermus aquaticus, right? Thermus aquaticus is found in hydrothermal vents, okay? And there are only few hydrothermal vents. I mean, even there, it's not that this organism is in plenty. It is there, okay? So the question is, suppose you want to, purify Taq polymerase, you can't be going into the hydrothermal vent, getting the pure culture of Thermus aquaticus and extracting that, right? So instead, it's easier to clone the gene, the Taq polymerase gene, right, in E. coli and express it, okay? And so it is much easier. You are not going to go and what should I say? You're not going to disturb the thermus aquaticus, this thing. Okay, otherwise, just imagine if cloning was not there, only thermus aquaticus was the only thing, then, for all the PCRs that we do in the world, I think you will not have enough thermus aquaticus, unless somebody, grows them artificially, which I think has not been that successful, right? Which has not been that successful. And so it is easier to clone any gene in E. coli and express them. Okay. E. coli is also considered as generally regarded as safe. E. coli is considered as a safe microorganism to work with, unlike something like Bacillus anthracis, which causes anthrax bacteria. Would you like to work with anthrax bacteria? No, people will not. You need to have special training, you need to have special facilities, you need to have special infrastructure. But unlike these, with E. coli, with a laminar flow hood and a clean room, you will be able to start working with E. coli. Okay. And E. coli again is one of the most thoroughly studied microorganism for which many tools are available for genetic manipulation. There are several plasmids that are available. You can do, if you want to do sequencing, whether you want to do, mutagenic studies, or whether you want to do express, all these things, you have lots of plasmids and other tools that are available for E. coli. That's why many people, they will start out expressing the foreign genes in E. coli. Okay. So we will, the subsequent slides are taken from this scientist called Marco Hyoven, who works in the crystallography and bioinformatics groups in the UK. Okay. So he has I saw some of his lectures and slides. They look very nice, appropriate for us. So I've decided to take it up. Okay, so all the acknowledgements to Marco Hyoven. Okay. And so the question is why express in E. coli? Because it's a well-established system. It's easy to manipulate. You have a large variety of vectors. You have many different kinds of E. coli strains. Okay. And you have many different methods by which you can introduce your foreign gene. It's low tech, right? As I said, you don't need to have a very high tech facility, like Bacillus. All you need is a laminar flow hood and a clean room. You can start working. It's safe. Most important thing is it is safe to work with E. coli, and it is inexpensive to grow. All you can do is take an LB broth and E. coli will start growing happily. Okay. And it is suitable for a variety of radioactive labelings. Like, suppose you want to do NMR, protein NMR, then, it can incorporate 13C, 15N, or, deuterium isotopes. All those things can be, incorporated. It will take it up, and it can also take up non-natural amino acids, which are very, very useful for crystallographic studies. Okay. And most importantly, the post-translational modification that you would see with mammalian cells are missing, right? Things like glycosylation, disulfide bridges, all these things, proteolytic processing, these are all missing. And this could be a blessing under certain circumstances, not under all circumstances, but under certain circumstances. But unfortunately, sometimes, when E. coli expresses a lot of these proteins, they clump together. They clump together and they become what is called as insoluble aggregates. Okay. And so you have an issue of solubility. Okay. And some of this can be sorted out with, when you have chaperones. I'm not sure whether you've been exposed to this concept of chaperones. Chaperones are nothing but, molecular proteins that are responsible for correct folding and sorting of proteins in the cells. Okay, so eukaryotes have a lot of chaperones. Okay, sometimes people have tried to express eukaryotic chaperones and eukaryotic proteins in E. coli, and they have been partly successful, not always, but partly successful. Okay, and depending on which system you use, we will discuss one system today. Depending on the level of and the amount of expression, okay, you can play around with the conditions for expression. For example, as we saw in the cloning strategy, you will use IPTG, isopropyl beta D thiogalactopyranoside for inducing the expression. Okay, so sometimes people use different concentrations of IPTG. Sometimes people use different temperatures to express genes of interest. Okay, so these are all some things that people use. So prokaryotic expression vectors come in many different flavors and forms. I'm just going to show you three. Okay, but you can imagine there are several new systems that keep on coming. I'm just showing you three. Okay, so this is the PET vector that we saw in the initial class when we were comparing cloning vector and expression vector. So this is a T7 system, and this is the multiple cloning site where we will be, putting our gene of interest. Okay, and this we will spend some time today on what this PET system is. Okay, P expression and T stands for T7. We will come back and spend some time on that. Okay. Just hold on for a minute. Hello? Sanjini. Three thirty. I'm in a class. I will let you know. 3:30 ko, okay? Okay. Yeah. So this is an expression vector which is commonly employed in almost all the laboratories. Okay. And 21A, as you can see, when we were students, there used to be PET2, PET3, PET4, PET5. It has now gone all the way to 37, 38, 39, something like that. So this is somewhere in the middle. Okay. So you can imagine PET has become a common this thing. It is now sold commercially by a company called Novozymes. Okay, and we'll come back to this in a few minutes. Okay, then you have another vector. Okay, now this is called as PJX. Okay, and the beauty of this system is that you can actually see that there are two things that I want to point out in this. One is that this has a promoter which is called, so in this case this was T7 promoter which drives this your gene of interest. Okay, in this case you have a promoter called PTAC. TAC is a hybrid promoter between tryptophan promoter and a LAC promoter. Okay, so this is a hybrid promoter, and then this promoter drives a gene called glutathione S transferase, and then you will be cloning your gene of interest downstream of this. So essentially, you are going to get a protein which is a, what should I say? It is going to be a fusion protein. It is going to be a fusion protein, fusion between glutathione S transferase and your protein. Okay. Obviously, when you want to study the structure and function of your protein, you don't want glutathione S transferase to be there. So you can, by the use of certain proteases, okay, you can very selectively cleave, okay, this fusion protein so that this glutathione S transferase goes away and your protein of interest will be purified entirely. Okay. There are ways by which we can do this. We will discuss in subsequent classes. Then you have another protein called PMAL, which is a maltose binding protein. It's a fusion between maltose binding protein. So you can actually, the multiple cloning site is somewhere here. Okay, and this comes as a fusion, just as the previous one was fusion between glutathione S transferase and your gene of interest. In this case, it will be maltose binding protein and your gene of interest. Okay, so we will be able to express these things in E. coli, and you will be able to express them at a fairly high levels. Okay, it all depends on the promoter. In this case, for example, if you look at this is driven by T7 promoter, right? This is driven by PTAC promoter, which is tryptophan plus LAC promoter, hybrid of that. In this case, it is the MAL promoter, okay, which is driving the expression of your foreign gene of interest. Okay, so three different vectors, three different promoters. driving the foreign gene of interest. Did I make it clear?
Yes, that's clear.
Okay. Okay. So if you look at you have a variety of vectors when it comes to expressing in E. coli. Okay, the native promoters are lag promoter, trip promoter, then this hybrid promoter called tag promoter, which is tryptophan plus lag. You also have trip promoters. Okay, and then you have arabinose promoters. Okay. And then you have certain viral promoters, but which are recognized by E. coli. For example, lambda is a phage that infects E. coli. Okay. And then in this lambda phage, you have two promoters. One is called as the left promoter and one is called as the right promoter. And then you have another phage called T5. Okay. T5 also infects E. coli and the promoter from T5 is recognized by E. coli. Okay. And then you have an unusual virus called T7. Okay. So you have T1, T3, T5, T7. Okay. So these are called as T odd phages. Okay. And this T7 phage, encodes its own RNA polymerase. And this RNA polymerase is an extremely what should I say, highly processive RNA polymerase. What is a processive RNA polymerase? Once it binds, it transcribes and transcribes and transcribes and transcribes and keeps on transcribing. Okay, that's the meaning of a highly processive RNA polymerase. Then you have tryptophan promoter, which is responsible for tryptophan biosynthetic enzymes. You can actually, when you have high levels of tryptophan, the promoter gets repressed. And unlike your lactose, where we do induction with IPTG, here induction is done by a small molecule called indole 2 acrylic acid. Okay. And as I said, lac-operon, we are all very familiar by now. Okay, lac-operon is repressed by the LAC-I gene, which binds to downstream of the promoter, and it is regulated by lactose or its analogs like IPTG, right? And it's a beautiful system. Lac-operon is something that is we are well versed with. Okay, you have synthetic promoters, which is you have trip and lac promoter. They have been combined, and the feature or the strength of this synthetic promoter is much better than your this thing, your native promoter. Ma'am, 3:30. ठीक है मैम, ठीक है मैम. No problem, no problem. Okay. ठीक है, okay. Thank you. Okay. So this promoter, the -35 is from the trip region, and the -10 is from the lac region. Okay. And typically this is again regulated by the lac system. So you can induce by IPTG. And it was shown to be much stronger than either of the parent promoters. Okay. And this is typically what is found in PJEX and PMAL promoters. Okay. Okay, so this is the this thing that I was about to tell this. So this is all T7 is an unusual bacteriophage. Okay, and the promoter of this gene 1 of the bacteriophage T7 is recognized only by the T7 RNA polymerase. Okay. And as I said, this is a highly fast and highly processive enzyme. Once this RNA polymerase latches onto a promoter that it recognizes, it doesn't recognize every promoter. Okay, it doesn't recognize E. coli promoter at all. T7 RNA polymerase recognizes only T7 genes, and it has a specific structure. Okay, and so it generally doesn't recognize any T7 promoter. But if you were to put any gene under the T7 promoter, this RNA polymerase will recognize and will make lots and lots of copies. Okay, so this was originally developed by a scientist called William Studier in the late 80s, and it has become the most popular expression as on date. Okay, In fact, every graduate student in Amity, not only Amity, I think in every other academic institution, when they want to express any foreign gene in E. coli, they will invariably end up using this PET system. Okay, and this company called Novagen sells the PET system commercially, and they have tens and tens of many different vectors, some with fusion protein, some without fusion protein, some they target the periplasmic space of E. coli, some that secrete the protein outside of E. coli. You know, they have many different vectors. Okay, maybe in one of the classes I will show you their catalog. Then you will know the extent to which, you know, this technology has been exploited for commercial purpose. So you see at the end of all your BSc or MSc, what do you want to do? You want to use that knowledge and make money out of it. I'm sure you guys will be doing IPR and this thing, right? In this semester, I guess, IPR and regulatory issues, either in this semester or next semester you will be doing, okay, which talks about intellectual property rights. You know, where do you derive this intellectual property right? You have been given some information, you exploit that to create a new knowledge, okay, which others don't know, and then you exploit that knowledge or you monetize that knowledge. You monetize that knowledge to make money out of it. Okay, it is one thing is, some people try to monetize it. Some people, they may not monetize it the way other commercial companies may do it, but at the end of it there is some monetization involved. Okay, so this T7 RNA polymerase can be inhibited by T7 lysozyme. Okay, so it makes sure that, that T7 also makes lysozyme, which actually is involved in the permeabilization of E. coli, and that's how the T7 completes its life cycle. We are not interested in T7 life cycle, so I'm not going to go into the details of it, but suffice to say that two things from T7. One is T7 RNA polymerase, which is highly processive, and people use it for doing this. And then you have T7 lysozyme, which has this ability to go and bind to T7 RNA polymerase and prevents it from the transcription process. Okay. Now the vectors that are used for T7 expression system, they are generally combined with the lac regulator and lac repressor gene to provide a tight regulation of this T7 expression system. Okay. And then at the end of it, the gene is generally, has a T7 transcriptional terminator. So this RNA polymerase recognizes only the T7 transcription terminator. You want to make sure that your transcription terminates at some point, right? And so it is generally done with the T7 transcription terminator. Okay. So this just shows you how the system looks like before induction. So this T7 induction is all done in specialized E. coli cells. It is not done in DH5 alpha, and it is not done in X7 blue. It is not done in JM101, whatever. These are all done in very specialized E. coli cells, which we will have a brief look in our next class. Okay. And these are called as BL21BE3, or sometimes people simply call it as BL21. Okay. So this is a specially engineered bacteria, okay, that carries the T7 RNA polymerase gene, okay, along with certain things in the E. coli genome. See, E. coli should not be having T7 stuff, but as I said, this is a genetically engineered E. coli which carries the T7 RNA polymerase gene in it. The upstream of this, is the lac UV5 promoter, which is a synthetic promoter, okay, which is followed by the lac operator, followed by the ribosome binding site, and then you have the T7 RNA polymerase. Okay. Now this E. coli RNA polymerase tries to transcribe it, okay, but no, this, as long as there is no lactose under normal conditions, your lac operator goes and binds this region, so it prevents the transcription. Okay, it prevents the transcription. Okay, and even if there is a small amount of T7 RNA polymerase is produced, this is effectively neutralized by the T7 lysozyme. Okay, so T7 lysozyme binds to this. Okay, and make sure that this is not expressed at all. Okay, this is not expressed. So this is the difference between, so there are two things that you can see here. One is this expression vector, okay, which is the PET system, and this is in the E. coli genome. So the E. coli genome has T7 RNA polymerase, and your gene of interest is cloned in the PET system. This is your expression vector, okay, and your expression vector has a T7 promoter, and so this T7 RNA polymerase needs to recognize this T7 promoter and drive high levels of your gene of expression, okay. So typically, you know, uh, under normal conditions, you know, when there is no induction with IPTG, right? When there is no induction with IPTG, the very small amounts of T7 RNA polymerase gets expressed, but that T7 RNA polymerase is essentially blocked by small amount of T7 lysozyme, which is present in these cells. These are, as I said, these are genetically engineered cells, and that prevents the T7 RNA polymerase from transcribing your gene of interest. Okay. Now what happens? So this is before induction. What happens after, when the T7 system is in action? Okay. So typically you will use IPTG for inducing this expression system. Okay. The moment you add IPTG to it, okay, this IPTG will go and bind to the repressor, right? And once it binds the repressor comes off. The E. coli RNA polymerase recognizes this LAC2V5 promoter and transcribes the T7 RNA polymerase, right? Now, once the T7 RNA polymerase is transcribed, you are going to get high copies of your T7 RNA polymerase. Okay, even if there are only few molecules of T7 RNA polymerase, it is enough to drive this system. Okay, so what happens is, once the T7 RNA polymerase is produced in enough numbers, okay, this essentially titrates out the T7 lysozyme, because T7 lysozyme is produced only in small amounts, small quantities, okay. That small quantity is sufficient when there is a basal transcription of T7 RNA polymerase. But under these conditions, you know, when you have used IPTG to induce this, you know, T7 RNA polymerase is produced in high quantity, and then this T7 RNA polymerase recognizes the T7 promoter, and then it starts expressing your gene of interest at very high levels, at, you know, fairly very high levels, okay. And generally for your routine studies, you know, for routine academic studies, this is a fairly good system to find out whether your gene of interest is getting expressed. And, you know, people typically, as I said, people do, you know, different concentrations of IPTG. People use these induction studies at different temperatures. Some proteins, you know, you can do at 37, they get expressed fantastic. Some proteins you need to go down to like 4 degrees centigrade, only then they will be expressing it. They'll be folding properly. And some proteins you need to, do at 25 degrees centigrade. So it all depends on the protein. Sometimes you may need to do, multiple different things before you optimize your expression system. In the next class, what I thought was, I will, show you how E. coli expression system has been commercialized, what all products are available. One typical product that is available is insulin. You know, it is even now, some of the recombinant insulin is produced in E. coli. Okay, there are many other therapeutic medicines, you know, which are produced in E. coli, which is used in the clinic today. Okay, so we will go and check that out in our next class. Any doubt that you may have until now, up till now? I started doing this for the maltose binding protein. Then I realized that I will spend some time on T7. In the next class, we will look at this BL21 E. coli cells, which are specialized E. coli cells. And then we will finish this T7 expression system. Then we will go to the other expression systems. Okay. Any questions?
No, sir.
No, sir.
Okay. These slides have not been mailed out. Okay, I'm still in the process of making it out. Okay, so once this set gets done, I will mail it to you. Okay.
Yes, sir.
Right. All right. All right. Okay. All right. So if there are no questions, then we'll meet on Friday then. All right. So take care. Stay safe. God bless you all.
Thank you.
Thank you, sir.
Thank you, sir.